Hey guys, welcome back to another episode of Honors Biology Room 500 with Mr. Mann. So what we opened up with to start class was the analyzing Chargaff's data. So if you want to look at that, go and fill that out for me, and then I'll go over that real quick. So the first couple things that we noticed, and this is kind of some things that other students said in class, was that A's and T's had about the same number, C's and G's had about the same number. They also said that A's and C, A plus C, and T plus G had around the same numbers as well. That all numbers equal to about 100% or 100. And so one of the things that we want to talk about first is Chargaff's A equals T and C equals G is very good for expectation. However, we're dealing with replication of DNA. And so when we do that, mistakes do happen. So notice that not all of the A's and T's are the exact same number, but they're pretty close. Same thing with the C's and G's. Number two, what we want to talk about is how do we find the value of the other numbers. So there was 35 for A's. And if we know that A equals T, that 35% is going to be with A, 35% is going to be with T, because they're equal to each other, based off Chargaff's base pair rules. Then what we're going to do is we have 30% left, because 100 minus 70 is 30. And we have two base pairs, and since they equal each other, they're going to be half of what the number is. So 100 minus 70 is 30. We're going to divide that by 2. We have 15% for C, 15% for G. And then what does that tell us? Well, a couple things it tells us. Basically what is going to happen is when we're replicating DNA, they tell us what the complementary DNA nitrogen base is, but also it could tell us what type of species it is or what type of organism based on the numbers of A's, T's, G's, and C's. If you look at your chart there on the different organisms have different percentages. So we're going to be able to answer what is the role of DNA or the role of enzymes in replication of DNA. How are leading and lagging strands synthesized differently? How does DNA replication compare in eukaryotes and prokaryotes? So what we're going to do first is talk about a little bit about enzymes. We're going to talk about these in a couple weeks. But basically, enzymes a protein, and a protein that goes through that takes a reactant of some sort and makes a product through a chemical reaction. So if you remember cellular respiration, photosynthesis, there was that arrow. The enzyme is the arrow. It takes something, comes in to this protein that fits what it's supposed to do. It changes it and makes a product that's a little bit different. And then we're going to talk about leading and lagging strands as we go through, and DNA replication compares eukaryotes and prokaryotes. So within this, there was a few theories: semi-conservative, conservative, and dispersive theory of how DNA replicated. So you see the two strands based on the key are the old strands to start with, and the new key, and then within the new key, the black is the old, the red is the new. And then semi-conservative, we see half black, half red on both. Conservative, it's all red, all black. And then dispersive, it's splices of black and red filtered through there. Okay, we use the semi-conservative model based on this study.
The experiment provides evidence for semi-conservative replication of the DNA molecule, where the two parent strands serve as the template for synthesis of new strands. In this experiment, bacterial cells were grown for several generations on a medium containing a heavy isotope of nitrogen, N15. The DNA in these cells therefore contained heavy N15 nitrogen. The cells were then transferred to a new medium containing the normal lighter isotope, N14. At various times after the transfer, samples of the bacteria were collected. The DNA was then extracted and dissolved in a solution of cesium chloride. The samples were then spun rapidly in a centrifuge. When the cesium chloride is centrifuged at high speed, a concentration gradient is established in the tube. DNA molecules move in the gradient until they reach a place where their density equals that of the cesium. DNA containing N14 moved to a position in the gradient determined by its density. DNA containing N15 is denser than that containing N14, so it sank to a lower position in the cesium gradient. After one generation in N14 medium, the bacteria yielded a single band of DNA with a density between that of N14 DNA and N15 DNA, indicating that only one strand of each duplex contained N15. After two generations in N14 medium, two bands were obtained, one of intermediate density, in which one of the strands contained N15, and one of low density, in which neither strand contained N15. Messelson and Stahl concluded that replication of the DNA duplex involves building new molecules by separating parent strands and then adding new nucleotides to form the complementary strand on each of these templates.
So that is our scientific study of how we found out that we use the semi-conservative model. So DNA replication is the process of which DNA is copied in a cell before the nucleus divides. And so this is kind of going back to what we talked about before Chris's break. So what stage is that? Where do we copy DNA before a cell divides? So if you said that DNA was copied in the S strand or in the S stage or the synthesis stage, you would be correct. Now, the single strand of DNA serves as a template for the new strand, as you saw in the video. Also, the rules of base pairing direct replication. So if I have an A, I'm going to pair it with a T. If I have a G, it's going to be paired with a C, and so on and so forth. And each body cell gets a complete set of identical DNA. Now, some important enzymes that we need to understand are these right here. Now, with enzymes, if it ends in ASE, know that it's an enzyme. So helicase, it's going to separate two strands of DNA by breaking the hydrogen bonds between the strands. It's going to unzip the DNA. Okay? DNA polymerase is going to catalyze and synthesize of DNA. It's basically going to add new free-floating nucleotides in the 5' to 3' direction. Also, it's going to proofread the new DNA sequence for errors. Ligase is going to catalyze the bond formation between Okazaki fragments. Another important word is what an Okazaki fragment is, and short segments of DNA formed on the lagging strand. So the existing DNA strand serves as a template, and the helicase and proteins are going to do the actual work of replication. And for a reminder, helicase is going to unzip, while free-floating nucleotides are going to form hydrogen bonds with DNA polymerase. As we see here, DNA replication or DNA helicase is going to unzip the molecule in both directions. Now, here's our picture. So DNA replication occurs in different directions on each strand. While replication on the leading strand occurs normally, replication on the lagging strand leads to the formation of Okazaki fragments of DNA. We should be able to label ligase, DNA polymerase, leading strand, lagging strand DNA, and our 5' 3'. So you will get the same exact picture, and it will be drag and drop for our quiz on your virtual day next week. So whether that be Wednesday or Thursday, because we're going to do an activity in class on Tuesday and Wednesday, and we're going to review the quiz on Zoom Monday. So what I'm underlining are the things that you need to know for DNA replication, for our replication fork, and then we're going to talk about it. So as you see here on the bottom, we have our leading strand template, and this is going to be our leading strand with our DNA polymerase. Behind it is going to be our DNA that the polymerase has already combined those hydrogen bonds, and it's going to follow the helicase through. While our lagging strand, or the top strand, is going backwards. So it's like walking backwards, so it needs a little bit of help. So we're still going to use our DNA polymerase, and it is going to still bind those hydrogen bonds. However, we have Okazaki fragments, or small sections of DNA, that we need to be able to bind together. And that's what our DNA ligase is going to be able to do. It's going to be able to help us with those nucleotides and bring them together and form the Okazaki fragment by binding those. And then, of course, we have our 5. It's going to go from our 3' to 5' end, our 3' to 5' end. And then over here we have our parental DNA that we are unzipping in order to make this copy within the leading and lagging strand. Now, there are multiple of these. This is a snapshot in time. So you're going to have multiple DNA polymerases, multiple helicases. And so once it gets to the point where there's DNA already made, the polymerase is done with its job. So then it's going to not perform its job anymore. The free nucleotides aren't going to keep adding and adding and adding and adding. Once it hits where DNA is already covered, then it's good. Now, these fragments require the function of the DNA ligase to join together short strands. Now, we're going to do this together real quick. So we have our three prime down here on our leading strand, 5 prime, and since this is anti-parallel, it's going to be opposite numbers. There's my polymerase here, my DNA polymerase. I put DNA poly. Do the same thing down here. And you're going to have to do a drag and drop of the picture that I showed you earlier. And my DNA back here I've already made. This is my parental DNA. I'm going to put pDNA. This is where my helicase goes. And I have my ligase here. And I have my Okazaki fragments here. I'm going to put OF. All right. And that's what you have to be able to label through a drag and drop on our quiz next week, as well as our test on the 26th/27th. So DNA replication begins at origin points. There are multiple origins, replication in eukaryotic cells, because it is not a shape of some sort. There is no start and end that come together. And so in order for this to be able to replicate, we have to have multiple origin points across the board in order to separate these strains fully. And in prokaryotes, there's only one origin of replication on their circular chromosome because the start and end are together. So as we look here, there's a circle, helicase is going to come through, it's going to put up all the way up. It's going to basically have 2 circles here. As you see, they're making DNA here within this, within each circle. So this is their DNA, and they're basically making a whole new other DNA basically around it as well. And that's going to be it. So if you have any questions, please let me know. Make sure that you have every assignment that's turned in with the due dates. If you have any questions, just let me know, and I'll see you all next time, and have a great weekend.